ऋषभ जी प्लीज स्टार्ट सेशन।
चलिए ऋषभ बाद में बात करेंगे अपने।
Good morning to all of you. Hello to everyone joining today's webinar. I am Rishabh Banod, and as a co-convener of this event, I am delighted to welcome a diverse and engaged audience. Your participation in this event, I am delighted to welcome a diverse, engaged audience. Your participation greatly enriches our discussion and underscores our shared commitment to advancing in our respective fields. I look forward to productive conversation and collective learning as we embark on this educational journey together. SPSR is distinguished for its engaging webinars, further enhancing its status as a leading platform for a continuous professional development in the pharmaceutical sector. I'm confident that today's session will upload about our tradition of excellence, providing valuable insights from the respective speaker. Let us seize this chance to gain a new knowledge, participate actively, and progress our field. Welcome, everyone. I look forward to a productive experience for all participants. Thank you for joining us. We are gathered here today for the commemorate the 94th SPSR webinar on nanotechnology-based colon-targeted drug delivery, a new paradigm in a pharma research, a distinguished event hosted by a Society of Pharmaceutical Sciences and Research, formerly known as SPSR, under the esteemed guidance of Dr. Shashi Alok Sir, a national president of SPSR. The society continues to play a pivotal role in fostering innovation and advancement in the field of pharmaceutical sciences. The SPSR is devoted to advancing the pharmaceutical field through a variety of strategies. Our mission is to organize conferences that support pharmacy-related initiatives and endorse programs tackling public health issues in India, focusing particularly on disease prevention. We make it a priority to recognize and honor pharmacists whose notable contributions drive progress in our field. At the heart of our mission is a commitment to spreading pharmaceutical knowledge, ensuring a steady and robust exchange of information about pharmaceutical practices and services. By striving toward these objectives, we aim to elevate a quality and impact of pharmacy profession, fostering a proactive and collaborative community dedicated to excellence. It's our great privilege to introduce Professor Sanjay Kumar Jain, sir, the Senior Professor of Pharmaceutics, former Head, Department of Pharmaceutical Sciences, Dr. Harisingh Gour, University, Sagar, Madhya Pradesh, as our distinguished speaker. Professor Jain's expertise greatly contribute to our event, providing substantial knowledge and insights. His participation in certain to enrich our discussion and exploration of nanotechnology-based colon-targeted drug delivery. We are grateful for his engagement and anticipate an enlightening session. It's our great privilege to introduce Mrs. Monika Sabharwal ma'am as a convener of today's webinar, who also serves as a founder of a National Secretary of SPSR and CEO of SPSR Periodicals Private Limited. Her significant involvement and a leadership in the pharmaceutical sciences and research communities are indicative of her commitment to this field. We are grateful to have her guidance in today's session. I am very much pleased to invite Mrs. Monika Sabharwal, ma'am, to warmly welcome our distinguished guest speaker, Professor Sanjay Kumar Jain, sir. Over to you, ma'am. Ma'am.
Thank you so much, Mr. Rishabh Banod. Hello, everyone. I am Monika Sabharwal, the convener of today's session, and it gives me immense pleasure to welcome you all to 94th webinar organized by Society of Pharmaceutical Sciences and Research SPSR. Today's webinar focuses on a very cut-edge topic in pharmaceutical sciences, nanotechnology-based colon-targeted drug delivery, a new paradigm in pharma research. Colon-targeted drug delivery is an area of very clinical importance, especially in the treatment diseases like inflammatory bowel disease, colorectal cancer, and the parasitic infections. Traditional delivery systems often fail to deliver drugs precisely to the colon, resulting in reduced therapeutic efficacy and increased side effects. The integration of nanotechnology in this area has opened a new realm of precision, efficiency, and targeted therapy, offering a transformating approach in the development of novel pharmaceutical formulations. We are truly honored to have with us today an esteemed speaker, Professor Sanjay Jain, sir, Senior Professor of Pharmaceuticals and former Head, Department of Pharmaceutical Sciences, Dr. Harisingh Gour University, Sagar, Madhya Pradesh, India. Professor Jain, sir, is renowned for his extensive research in targeted drug delivery systems and nanotechnology-based pharmaceutical innovations. His expertise will undoubtedly provide us with the valuable insights and broaden our understanding of this highly relevant and evolving area of research. As we celebrate 94th edition of our webinar series, I will also like to take a moment to highlight some key achievements of SPSR. We are proud to have built a global community of pharmaceutical professionals and researchers, with our webinars reaching participants of over 25 countries. One of the most encouraging milestones is the balanced representation among the SPSR members. 50% males and 50% female professionals, showcasing our commitment to gender equality and exclusive professional growth. Our digital footprint continues to expand rapidly. The SPSR WhatsApp channel has now over 15,000 followers, and our LinkedIn network has grown to more than 7,200 professionals. These numbers reflect the global interest and enthusiasm we have earned through the consistent efforts to disseminate knowledge and foster collaboration. All our webinars are completely free of cost, making expert-level education accessible to students, researchers, and professionals alike. In addition, participants receive certificates that contribute to their academic and professional development. We continue to receive an overwhelming positive feedback, with more than 99% of our participants reporting that SPSR webinars have helped them to grow in their professional journey. We are immensely grateful to our members, followers, and speakers who make this ongoing success possible. Before we commence, I would like to extend a very warm welcome to our National President, Dr. Shashi Alok, whose visionary leadership has been crucial in the impressive transformation of the SPSR. I would like to extend very heartfelt thanks to the significant contributions made by Mr. Rishabh Bhanot, the co-convener of this webinar, and organizing committee members, Dr. Pradeep Golani, Dr. Saurabh Kose, Dr. Sanjay Nagdev, Dr. Ankur Agarwal, and Mr. Junaid Tantre. Their tireless efforts have played a very crucial role in transforming the vision of this event into a reality. Join the SPSR Vibrant Committee of 57,000 pharma professionals actively engaging across LinkedIn, YouTube, Facebook, and WhatsApp. Let's connect, learn, and grow with the top-tier webinars and the exclusive content every time. Do not miss out this unparalleled opportunity to elevate your pharmacy practice. Stay updated by subscribing to SPSR YouTube channel and enabling the notification bell icon for instant alerts every time. Follow our journey in advancing pharmaceutical sciences that will benefit the society overall. With that, I once again extend a very warm welcome to all of you and express my sincere thanks for your continuous support every time. Please join me in welcoming the Professor S.K. Jain sir as we begin today's scientific session and one of the most promising areas in the pharmaceutical research. Your enthusiasm, active participation, and trust in SPSR drive us to keep going strong and strong. Thank you, and let's begin the 94th SPSR webinar. Thank you. Over to you, Mr. Rishabh Banod.
Thank you, ma'am. I highly appreciated Mrs. Monika Sabharwal, ma'am, granting me the opportunity to present this webinar. And now let me take the privilege to start with a short introduction of our esteemed guest speaker, Professor Dr. Sanjay Kumar Jain, sir. He is a retired senior professor of the Pharmaceutics from the Department of Pharmaceutical Sciences, Dr. Harisingh Gour, Central University, Sagar, Madhya Pradesh. During the active services, he was also holding a position of Directorate, Planning and Resource Generation and Chairman, Council of Industry Higher Education, collaboration of Dr. Harisingh, Gour University, Sagar, Madhya Pradesh. He is a former head of Department of Pharmaceutical Sciences and also former Dean of School of Engineering and Technology, Dr. H.S. Gour Vishwavidhalaya, Sagar, Madhya Pradesh. With over 35 years of experience in teaching and research and 2 years in industry, he has more than 220 research publications with more than 60 H-index and 320 I-10 with over 18,000 citation index. He has also written books and more than 50 book chapters in different national and foreign references books. He has 7 patents awarding, including 1 US and 1 European patents, 1 African patent, and as well as 4 Indian patents to his credit, and also has filed 3 more Indian patents. His main research area is designing and development of various new drug delivery systems for the treatment of disease associated with GIT tract and the brain. Professor Sanjay sir has maintained high standards of research and teaching throughout his service to the university. In 2018, he has been conferred with the India's most prestigious and highest award, Visitors Awards 2018, also known as President Award 2018 by His Excellency Shri Ramnath Kovind ji, Honorable President of India at Rashtrapati Bhavan, New Delhi on 2nd May 2018 for his excellent research in basic and applied sciences. He has also been ranked 4 times consequently under 2% scientist by the Stanford University, USA in 2020, 2021, 2023, and 2024. Professor Jain sir has also been awarded thrice in 1992, 2003, and 2006 with IDMA Award for the Best Research Publications. Besides this, he has been conferred with the prestigious MLR Khurana Award 2003, CRS Capsugel Pfizer Innovation Award USA 2004, Best Research Award in Pharmaceutical Technology in 2005, Award of Research Excellency by the European Institute of Scientific Research, Netherlands, National Teacher Excellence Award in 2012 by Honorable Governor of Madhya Pradesh, and Pharma Recognition Award in 2021 by Pharmalok and Nirali Publisher, New Delhi, as well as Lifetime Achievement Award 2019 for his excellence in teaching and research. The Department of Science and Technology, the Government of India, has recorded Professor Sanjay sir as a ranked 3rd National Citation Repository 2016 under the subject Pharmacology and Pharmaceutics. In 2021, the Association of Pharmaceutical Teachers of India, APTI, has conferred a most prestigious award, Dr. Mrs. Manjushri Paul Memorial Award for the Best Pharmaceutical Scientist in 2021. Professor Jain sir has successfully guided 32 PhD and more than 160 m.pharm dissertations with, as well as has completed 8 major research projects funded by the various national agencies. Dr. Jain sir has presented a number of research papers in various national and international conferences and delivered many invited lectures in various conferences on different topics. He is a member of editorial board of Citation Review, Critical Review in the Therapeutic Drug Carrier System, Beagle House, UK. regional, regional editor of Current Signal Transduction Therapy, and Bentham Science publisher and Bentham Science brand ambassador. He is also a referee of various national and international journals of repute. He has also been chairman of and member of universities, various committees and board of studies of various universities. He is also a UGC nominated member of governing bodies of Bombay College of Pharmacy, Mumbai, and ISF College of Pharmacy, Moga, Punjab, as well as the member of University Advisory Board of Sage University, Bhopal, Madhya Pradesh, and a member of Scientific Advisory Committee of National Anti-Doping Testing Lab, New Delhi. He is one of the members of Board of Management of NIPER Ahmedabad. He is a lifetime member of many learned societies and he has been organized many training and programs and conferences. It is great satisfaction and pride that we declare that we have exceeded 1400 registration for this webinar, and registration received from the India, Albania, Bahrain, Baku, Bangladesh, Egypt, Ethiopia, Indonesia, Iraq, Kenya, USA, Kuwait, Malaysia, Mexico, Myanmar, Nepal, Nigeria, Oman, Philippines, Romania, South Africa, Syria, Sri Lanka, Turkey, UAE, USA, and Vietnam. We are deeply moved by the substantial and varied response we have gained. Our heartfelt gratitude is extended to each person who has taken a moment to participate in this significant event. Thank you for all of us joined today. Let's make this webinar an insightful and enriching experience for everyone involved. We have our esteemed keynote speaker, Professor Sanjay Kumar Jain, sir, with us. Welcoming, sir. Your insight would highly be valuable, and I would appreciate if you could present them in a speech. And over to you, sir.
Thank you very much, Dr. Rishabh Banot, for giving us such a elaborated introduction of mine. I'm really honored to speak in this 94th webinar that is that is organized by the Society of Pharmaceutical Sciences and Research. And this is a well-known society of the pharmacy professionals. And I'm really thankful to Dr. Shashi Alok for giving me a chance to speak among the so many pharmacy professionals. Today I shall be delivering a lecture on the nanotechnology based specific colon targeted drug delivery systems. So I will be discussing the salient features of the colon specific drug delivery systems and how it is beneficial for further research and different aspect, nanotechnological aspect of the colon drug delivery system. So if you permit me to, I can share my slides.
Yes, sir. Go ahead, sir.
I'm audible. My audio is clear.
Yes, indeed, sir. Yes, sir. Slide is working. You can continue, sir.
Just a minute. Just a minute. Just I'm adjusting the all the these symbols. No? So, well, as you know, I shall be talking about the nanotechnology-based colon targeted drug delivery system, a new paradigm in pharmaceutical research. And as we are very well aware about the requirement of the parameters for the development of any drug delivery systems. If we recall our knowledge, then it is very clear that for the development of any drug delivery system, you will require knowledge of the chemistry, biology, and material science. Because for the preparation of any pharmaceutical product, effective product, safe and effective product, you will require the drug molecule and that to be incorporated into any substrate, that is a material, a net material, and it should be delivered to the any biological system in such a way that it will show some beneficial effects that will be beneficial to the human being or the patient, right? So you should have knowledge of this uh, chemistry, biology, and material science. Say, if you have a knowledge of molecular process of life, allowing us to manipulate and modulate living organisms in beneficial and sustainable way, that will be very beneficial. And using that knowledge, we can manipulate our drug delivery system so that we can effectively deliver the drug to the disease organ, and we can reduce the side effect of the drug. If you see the dynamics of the drug in the body, and then you will find that when you administer any drug, it will, if it is having the first order absorption, then drug has to cross the biological membrane, that it will reach to the blood circulation, and through the blood circulation, it will be distributed to different organs of the body if it is not target specific, if it is not disease specific. So for if it is the disease specific, then after reaching to the blood circulation, the delivery system will directly reach to the your disease organ and release the drug and will get the effect. But if it is in the form of the conventional dosage form, then it will reach to the blood circulation. Drug will reach to the blood circulation after the absorption of the drug from the absorption site. Then it will be distributed to different parts of the body. So the concentration of the drug in the body will be gradually increasing as on increasing the absorption from the absorption site. Then when drug will absorb, we will not get the, we will not be able to see the effect, but we will be see the effect of the drug when the drug concentration in the body is reaching above the minimum effective concentration. The concentration, minimum concentration required to get the effect of that drug. So we'll get the minimum, we get the effect of the drug when when the drug concentration will increase, or we can say that when the drug concentration will be above the minimum effective concentration, right? Now, duration of the action depends up to what time drug concentration will remain above the minimum effective concentration. So that will decide the duration of action of the drug, and when the drug concentration will come down the minimum effective concentration, then we will not be able to see any effect of the drug. But same time, due to the absorption, continuous absorption of the drug concentration in the body will be increasing, and sometimes if your dosage form or drug delivery system is not properly designed, then drug concentration may exceed the maximum safe concentration. If the drug concentration in the body is increased above the safe concentration, will get the side effect of that drug or toxic effect of the drug along with the beneficial effect of the drug. So being the pharmacy scientist or formulation scientist, our intention is to design such delivery system which will keep drug concentration in this therapeutic window. That means above the minimum of concentration and below the maximum safe level. You are required to keep the concentration in this therapeutic window for a longer period of time so that you will get a response, a beneficial response of the drug for a longer period of time after the administration of the drug. We have to same time we have to design such delivery system which will show faster onset of action after the absorption of the drug. If the drug is immediately, if the drug concentration in the body is crossing the minimum effective concentration, the onset of action will be reduced, right? So immediately we'll get the response of the drug. So it depends on the how the drug will be reaching to the blood circulation and how the drug is reaching to the disease organ, so that will get the response of the drug, whether you will get the beneficial response or the some side effect of the drug. It depends how the drug is absorbed, how much drug is absorbed, how much drug is reaching, and how the drug is reaching to the target or disease organs. So being the pharmacy scientists, we must have to formulate such delivery system which will give faster onset of action and longer duration of action, and that will keep drug concentration in the therapeutic window for a longer period of time. This will be our intention for designing a suitable drug delivery system, or we can say for designing the safe and effective drug delivery system. These are the challenges we face during the designing of the drug delivery system for the safe and effective drug delivery system. First challenge that we face, that is the prevention of the drug from biological degradation. What happens when we take the drug, say we take the drug through the oral route, drug may degrade in the gastrointestinal lumen due to the change in the pH, due to the release of the so many enzymes, so interaction with the so many enzymes, drug may degrade, or drug may degrade in the liver after the absorption of the drug. As you know, the liver is the prime organ for the metabolism of the drug. So that drug may degrade. Our intention is to design such delivery system that will prevent this degradation or prevent degradation of the drug in the biological system, so that effective undegraded drug will reach to the disease organ or in the body. Keeping drug concentration in the therapeutic window. This is another challenge for the pharmacist who are designing the drug delivery system. He has to keep drug concentration in the therapeutic window and should reach this. He has to design such delivery system that will cross this minimum effective concentration in a shorter time. So this is, and it will keep drug concentration in this therapeutic window for a longer period of time. So this is another challenge. Another challenge is product half-life extension. That is related with the shelf-life of the product. As we know that the cost of the product is also depends on the shelf-life of the product. If the shelf-life of the product is less, then cost of the product will increase. In this case, the return from the market, return of the product from the market will be more if the shelf life is more. So our intention is to keep drug product shelf life longer, so that the cost can be reduced. The return from the product is can be minimized. Another challenge which we face, that maximize utilization of the delivered drug. That is the whatever drug, whatever amount of the drug that you deliver to the biological system or to the patient or human being, and if the whole amount, a whole delivery amount is utilized for showing the therapeutic effect of the drug, then that will be the best delivery system or best system, best dosage form. So where the utilization of the drug is maximized is if you deliver say 100 milligram of the drug, only say 1 milligram or 100 microgram drug is utilized for showing the results. The remaining 99 milligram or 99.9 milligram drug is wastage. So by increasing the utilization of the drug by the target organ or disease organ, we can reduce the dose of the drug, and same time we can reduce the cost of the drug. Another one, this can be achieved by preparing the effective drug targeting. This is also a challenge for delivering whole the delivered drug, for releasing the whole delivered drug to the target organ. This is really a challenge. So we must have to design such delivery system which will effectively deliver to the target organ. It will not be drug will not be delivered from the drug delivery system to the non-unwanted organs of the body. Another challenge is patient compliance. Whatever the delivery system we prepare or design or develop, it should have the patient compliance. Without patient compliance, the development or designing of the drug delivery system will be useless. So because our prime user are the patient, and we must have to keep the comfort of the patient in mind during the designing of the drug delivery system. Last one is the making the drug delivery system cost effective, affordable drug delivery system we must have to design. And this is possible only if you have the integrated knowledge of chemistry, biology, and material science. So, today we'll discuss, because discussing in general about the best targeted drug delivery system is very difficult. So I choose the colon. I choose the colon as a organ for delivering the drug, because delivering drug to the colon is not easy task. If you take it through the oral route, it is drug has to travel from mouth cavity to the ileocecal region, right? Long way it has to travel. It has to face variation in the pH in the gastrointestinal lumen, as well as it has to face with the number of the enzymes which are released in the gastrointestinal lumen. And it may interact with the dietary components or GIT components also. So we must have to protect the drug all the way from mouth cavity or buccal cavity to the ileocecal region. And at the same time, if we deliver it through the rectal route, it is not easy route for or convenient route for delivering drug to the colon. Although delivering drug to the colon through the rectal route is required less time, because drug has to travel short distance. But it is not easy one. It is not convenient one. You will require expertise for delivering drug to the colon through the rectal route. Say, if you see the structure, because if you would develop or design any drug delivery system, you must have to know the biology, anatomy, physiology of that particular organ where you are delivering the drug. So colon is as you know, it is the lower part of the GIT. It extends from the ileocecal junction to the anus. Colon is upper 5 feet of the large intestine, and rectum is the lower 6 inches after the colon. And if you see the structure of the colonic membrane, then you will find that it is having number of the membranes, consist of number of membrane. In the colonic lumen, you will have the mucosal membrane, and the mucosal membrane of the colon is thicker than the mucosal thickness of the mucosal membrane of the upper intestine. When I say upper intestine, it means the small intestine and stomach. So the mucosal thickness of the mucosal membrane in stomach and small intestine is lesser than the thickness of the mucosal membrane in the colon. And this mucosal membrane is the producing main resistance for the delivering drug to transfer of the drug to the colonic lumen to the serosal side of the colon. And below the mucosal membrane, there is a submucosal membrane, and below that is circular muscular layer, longitudinal muscular layer, and serosal membrane. So if a drug has to cross all these membrane, then it will reach to the blood circulation. It's not easy task, and this colonic membrane, colonic epithelial membrane, is different from the epithelial membrane of the upper intestine, because the colonic membrane is having the space between the cells of the epithelial cells of the colonic membrane is very less. They are joined together by occluding junctional complex. There is a protein by which the cells are joined. So intercellular transmission is not possible by this colonic membrane. Otherwise, if you want to cross this membrane, you will require to change the permeability. You will require to use the permeability enhancer. Same time, our gastrointestinal membrane, which is consist of after the esophagus, the main part is the stomach, then small intestine, and then colon or large intestine and rectum. These are the four parts of our GIT membrane. So after the taking any meal which is coming into we are administered through the oral route, it will first come to the stomach, then it will go to small intestine, then the large intestine, and leave the body through the rectum. So in this passage, drug has to come across with the variation of the pH. The pH, if you see the pH of the stomach, that is about pH 1.5 and 2. And the length of the stomach is 20 centimeter. And residence time, any material which will remain in the stomach, that is for 1 to 3 hours. Then after 1 to 3 hours, the content of the stomach will transfer to the small intestine. And the length of the small intestine is approximately 730 centimeter. And the residence time is about 3 to 5 hours, means 3 to 5 hours. And the material is taking 3 to 5 hours to cross this small intestine. Small intestine is having 3 parts, that is duodenum, jejunum, and ileum. The pH now is gradually increasing from the acidic pH from 1.5 or 2.5 of the stomach. Then the pH of this is increasing from 2 to 4.5 to 5 in the duodenum. And in the jejunum, the pH is further increased 5 to 6.5, and ileum will have pH 6 to 7.5. Ileum is the last part of the small intestine, and then when material of the ileum transfer to the colon or large intestine, then it will reach to the ascending colon. First, it will reach through the cecum, it will reach to the ascending colon, where the pH is suddenly dropped. As soon as material will reach to the ascending colon, the pH of the ascending colon is dropped from 7.5 to 6.4. And the fall in the pH is due to the presence of short chain fatty acid arising from bacterial fermentation of polysaccharides, right? So the fermentation of, say, lactose to lactic acid, that will reduce, lactic acid will reduce the pH of the ascending colon. Then the material will, when we transfer from ascending colon to transfer colon, the pH is gradually again increase from 6.4 to 6.6, then descending colon to the pH again increase, that is 6.6 to 7, and sigmoid colon, last one is the sigmoid colon, where you will have pH about 7 to 7.5 or 8. And rectum, that is the last part of the gastrointestinal tract, which is having the length of 12 centimeter, and the pH is 7 to 8. And the residence time of the colon, if you see the residence time of the colon, the content in the colon will remain more than 30 hours, right? Or sometimes it is the residence increased to 48 hours, means that any material which is coming to the colon will remain in the colon up to 48 hours. So this is one of the advantage, one of the advantage where we can manipulate with the drug delivery systems, because drug delivery system will remain in the colon for more than 30 to 48 hours. So we can manipulate it, the drug delivery system. Say, if you see the this microflora, our colon is full of microorganisms. Say, upper part of the intestine, this is the unique feature of the gastrointestinal tract. Our gastrointestinal tract is having the number of beneficial bacteria which helps in digestion of the food, right? So upper intestine means small intestine, stomach, having the only 6 bacteria, right? That is the Clostridium, Mycococcus, Sarcina, E. coli, Klebsiella, and Proteus. So these 6 microorganisms are present in the upper part of the GIT. But if you see the microorganisms which are present in the colon or large intestine, there is a long list of the aerobic and anaerobic bacteria which are present in the this large intestine or colon. These microorganisms releases number of enzymes for the digestion of the undigested food which is coming into the colon, right? So 30% of dry weight of the feces consisting of bacteria. You see the population of the bacteria present in the colon, that is 10 to the power 11 to 10 to the power 12 colony forming unit per ml, as compared to the bacterial count in the stomach, that is 0 to 10 to power 3 colony forming units. So you can imagine the number of the bacteria present in the colon. It's a huge amount of the bacteria present in the colon. And because of these aerobic and non-aerobic bacteria, number of enzymes are releasing in the colon. They are some of the reducing enzymes, some of the hydrolytic enzymes. Reducing enzymes in nitroreductase, azoreductase, N-oxidase reductase, sulfoxide reductase, and hydrolytic enzyme esterases, amidases, glycosidases, gluconidases. These enzymes are released by the colonic bacteria in the colon, right? And because of release of these enzymes, number of metabolic reactions are carried out by the colonic bacteria. that is hydrolysis, reduction, decarboxylation, dealkylation, dehalogenation, deamination, acylation, esterification. These metabolic reactions are carried out by the bacteria present in the colon. So, well, in the designing of, we can choose material which are metabolized by the enzymes released by the exclusively released by the colonic bacteria. So we can make by selecting the material, such type of material which will metabolize or degrade in the colon by the colonic enzyme or enzyme released by the colonic bacteria. So due to this exclusive uh, enzyme which are present in the colon, we can trigger the release of the drug in the colon. Say in case of the azoreductase enzyme is a reducing enzyme and it reduces the azo bond, selectively it reduces the azo bond. It is acting on the azo bond. So if the material, if the polymer you select or synthesize, which is having the azo bond in the skeleton of the polymer, and if you select those polymer for the fabrication of the drug delivery system, that will not release drug in the upper part of the GIT. It will release drug in the colon only, because in the colon azoreductase enzyme will degrade the aromatic this azo bond of the aromatic polymer and release the drug. Similarly, polysaccharidase enzyme, this is also being released by the colonic bacteria. It is exclusively present in the colon. So polysaccharidase enzyme is responsible for the degradation of the polysaccharides. So we can use such type of the enzyme, and we can select suitable material which are degraded by these enzymes which are released exclusively in the colon. It is not really so easy to deliver drug to the systematic circulation from the colon, because as I told you, if you see the colonic membrane, it is having the number of layers, right? And because of that, transmission of the molecule, drug molecule or any content from the colonic lumen to the serosal site is difficult one. Similarly, apart from the colonic membrane, complex structure of the colonic membrane, there are other barriers also that produce hindrance in the absorption of the drug or the material from the colonic lumen. The small surface area of the colon. Colon is having very small surface area. If you compare it with the area of the small intestine, in the small intestine, because of presence of villi, microvilli, the area of the small intestine is many times larger than the area of the colon. So surface area of the colon is very limited, so absorption will be limited. And in the lumen of the colon, there is a specific and non-specific drug binding with the dietary components. And the dietary components and the bacterial release from the colon. So that will bind with the drug, and these sometimes are producing non-absorbable compounds. The high viscosity of colonic content is also impaired the diffusion of the drug from the lumen to the mucosa, right? The movement of the drug in the colonic lumen is restricted because of the high viscosity of the colonic content. The mucous membrane at the epithelial surface of the colon, as I told you, it is a thicker membrane, and it has the drug reaching to the epithelial surface. It produces hindrance. Unstirred water layer, this is the inic layer is present in the colonic membrane. Below the epithelial surface, there is a hydrophil water layer you will find in the colon that is produce barrier for the lipophilic drug. And the epithelial epithelial membrane of the colon is having the colonocyte and offloading junctional complex in the colonic epithelial layer, which provides physical barrier. And the last one is the epithelial surface, the lipid bilayer of producing hindrance for the transfer of the hydrophilic drug. So by this colon, only the absorption is very low. Only water and dissolved minerals are absorbed. Then, if you see this figure where I have tried to explain you the differences between the colon-targeted drug delivery system and the conventional drug delivery system, right? So, well, if the drug is delivered through the conventional dosage form, then most of the drug, delivered drug, is absorbed through the upper part of the GIT, that is from the small intestine and the stomach. So it will reach to the blood, and through the blood, drug will come into the colon if it is required for the treatment of the diseases associated with the colon. So only limited, very limited drug will transfer directly to the colon, which is unabsorbed in the upper part of the GIT. But if the drug is entrapped in the colon-specific drug delivery system, then maximum amount of the drug directly reaches to the colon or large intestine. Very limited, very less drug will absorb from the upper part of the GIT. So in this way, if you want to treat the disease associated with the colon, you will require to entrap drug in the colon-specific drug delivery system. So directly drug we can deliver to the colon. Because if it is delivered through the conventional dosage form or the general dosage form, like tablet or capsules, then most of the drug will be absorbed and you will get the side effect of that drug. There is an example. This is metronidazole. Metronidazole is used for the treatment of the amiasis, right? So, and for the treatment of amiasis, it is the dose is 500 milligram 3 or 4 times a day. You will require to take total 1.5 or 2 gram of the metronidazole in 24 hours, right? Why? And for the just for killing a tiny amoeba, chota sa amoeba hai, tiny amoeba, to kill that tiny amoeba, you will require 2 gram of the metronidazole or 1.5 milligram of metronidazole. It's really a challenging one. To kill that amoeba, only say few microgram of the metronidazole is required to kill the amoeba. So if we deliver few milligram of the drug directly to the colon, then the amoeba may be killed. Because in the conventional dosage form, the drug metronidazole, most of the delivered metronidazole is absorbed, and it will reach to the absorbed from the upper intestine and reach to the blood circulation, and from blood circulation it will come to the colon for the killing of the amoeba. And for if it is given through the colon-specific drug delivery system, then no metronidazole will absorb from the upper part of the GIT. All the administered drug will be delivered to the colon. It will be more effective one. So in this way, we can treat the amoeba and we can reduce the side effects of the drug. As you know, by this discussion, we come to know that colon is the potential site for the drug delivery system because it is having low hostile environment for the degradation of the drug and molecules and long colonic transit time, say 20 to 30 hours, minimum 20 to 30 hours. So we can manipulate with the drug release or designing of the drug delivery system. Unique microbial flora, as we have seen that colon is full of aerobic and anaerobic microorganisms, which releases a number of the enzymes. And using these enzymes, we can select suitable material for taking the release or delivering the release exclusively to the colon. It is a highly responsive to absorption enhancer. Because if you want to deliver drug to the systematic circulation from the colonic lumen, you will require to alter the permeability of the colonic membrane by using the some permeation enhancers. So the membrane or colonic membrane is highly responsive to the colonic membrane. So which drug candidate we can use for designing the colon-specific drug delivery systems? Those drugs which are having the poor bioavailability from the stomach and small intestine can be used for delivering it through to the colon. That is the peptides or the peptide drugs, or those drugs which are having the deleterious effect on the gastric mucosa, like non-steroidal anti-inflammatory drug, so that can be delivered to the colon, or drug required for the treatment of diseases associated with the colon. So we can like amiasis, colon cancer, some colonic ulcerative colitis or inflammatory bowel disease. So for the treatment, especially for the treatment of amiasis, colorectal cancer, ulcerative colitis, inflammatory bowel disease, the drug is required to deliver to exclusively deliver to the colon. So if it is giving through the conventional dosage form, then the toxicity of that drug will be very high. So it is required to deliver it through the colon-specific drug delivery system. And there is some candidates like for the colon-specific drug delivery system, some digestive enzyme supplementary that is for local action, and some 5-aminosalicylic acid derivatives for the treatment of irritable bowel disease, Crohn's disease, or irritable bowel syndrome, and that is for topical action, and the 5-fluorouracil that is for treatment of the colorectal cancer, and non-steroidal anti-inflammatory drug that will prevent the gastric irritation for systematic absorption. Some oral vaccine we can deliver through the colon, like typhoid vaccine, and insulin can be delivered through the colon. That is for oral delivery of the peptides. The colon-specific drug delivery system having some on oral administration, having some characteristics like releasing drug exclusively in the colon, it will increase the colonic bioavailability, enhance the therapeutic efficacy by localizing to the colon. It will reduce the systematic adverse effect by reducing the absorption of drug from the upper intestine. It will maximize the drug utilization. It will reduce the dose of the drug, maximizing the drug retention time in the colon. So these are the characteristics of the colon drug delivery system. And if you for delivering drug to specifically to the colon, number of stages are adopted. There are some emerging stages, especially for the delivering the drug delivery system, like emerging stages for colon targeting is product approach. There are certain type of the product like azobone product, dextran product, glycosidic product, these are the products which will exclusively release drug, parent drug in the colon by the because this whole drug will degrade in the colon, only in the colon by the presence of the suitable enzymes which are released by the colonic bacteria. Size depending the delivery system, that is a nanodelivery system that will, if you develop a nanodelivery system that will having a good EPR effect and will adhere to the membrane for more time, also the resident times can further be increased in the colon if it is designed in the nano size. Surface charge dependent drug delivery system, both cationic and anionic drug delivery system, both can be used for the colon specific drug delivery system. pH dependent drug delivery system like hydrazines, which are pH sensitive one. So by using the pH sensitive polymer, we can develop the colon specific drug delivery system. Biodegradable drug delivery system, which are degraded by the enzymes released by the colonic bacteria like pectin, guar gum, cytosine, chondroitin, something. There are a number of biodegradable polymers that can be used for the triggering the drug release in the colon. Redox drug delivery system, that is specifically for the inflamed bowel disease or irritable bowel disease where the colonic membrane is inflamed. In case of the inflamed colonic membrane, the reactive oxygen species ROS level is increased, and that increased ROS level can be used for triggering the drug release from the oxygen sensitive polymers like thioketone. Active targeting depending drug delivery system, this is the especially dual targeting concept where the drug is designed in such a way, drug delivery system is designed in such a way that will deliver drug to the colon, and then further the drug is designed in such a way that it will be carried to the disease organ like for the treatment of colorectal cancer, specifically for delivering drug to the colorectal cancer site. This active targeting dependent delivery system most suitable one. Nanocomposite based drug delivery system. This is using the say Montmorillite. I have used Montmorillite and prepared a cytosine layer composites. So by the use of the Montmorillite and cytosine, we prepare a layer composites, and this layer composites is very. In this layer composites, we have incorporated 5-salicylic acid mesylagine. And this nanocomposite is very effective for the treatment of the inflamed bowel disease, IBD. Dendrimer-based delivery system. In the skeleton of the dendrimer, if you put the azo bonds, right, instead of amide bond, if you put the azo bond, right, and double bond, and then in the skeleton of the dendrimers, then these dendrimers will be colon specific one. It will degrade only in the colon when these dendrimer come in contact with the come in contact with the azoreductase enzyme, which is excessively present in the colon. Time release system, yes, this is very most convenient system. And first time release system for colon delivery is pulsing cap. Pulsing cap is nothing but it is the here using this pulsing cap, the release of the drug from the drug delivery system or capsule is delayed up to the 7 hours. It is designed in such a way that it will delay the release up to the 7 hours by the time the drug delivery system or pulsing cap will reach to the colon. So, well, this is very first approach, azo bond approach. The very popular drug molecule that is used for the treatment of the inflammatory bowel disease. This is sulfasalazine. Sulfasalazine is nothing but it is the 5-amino salicylic acid coupled with the sulfapyridine, right? It is having one molecule of 5-amino salicylic acid and one molecule of sulfapyridine, right? And these are coupled with the azo bond, right? So this molecule, this sulfasalazine is non-absorbable in the upper part of the GIT. When it will reach to the colon, the azo reductase enzyme will be acting on this molecule, and it will cleave this azo bond and release the 5-amino salicylic, one molecule of 5-amino salicylic acid and one molecule of sulfapyridine. But sulfasalazine is having the toxicity side effects, and that is because of the release of the byproduct, that is the sulfapyridine in the colon. So you will have the side effect. But so to reduce this side effects, the sulfapyridine is replaced by some amino acids, right? So 5-amino salicylic acid is 5-amino salicylic acid is coupled with the conjugated with the this amino acid. Like if it is conjugated with the 4-amino benzoyl beta-alanine, the conjugate is called valsalazine, right? So, and it is conjugated with the azo bond, right? So when this valsalazine will come in contact with the azo-reductase enzyme, this azo bond will cleave and will get one molecule of 5-amino benzoyl beta-alanine, that is amino acids, the biocompatible one, and one molecule of the 5-amino salicylic acid. So this is another product of the 5-amino salicylic acid, where 4-amino benzoyl hippurate is used, and it is conjugated with the azo bond, with the help of azo bond with the 5-amino salicylic acid, right? And most say safe product is osalazine. Osalazine is nothing; it is having the two molecules of 5-amino salicylic acid joined together by the azo bond, right? So when this azo bond is cleaved in the presence of the azo-reductase enzyme in the colon, we'll get 2 molecules of the 5-amino salicylic acid. Another one is the glycoside prodrug. Glycoside, as you know, that polysaccharidase enzyme are released by the colonic microflora. One of them is the glycosidase. These are the polysaccharidase enzyme released by the colonic microflora. That is the beta D galactosidase, beta D glucosidase, alpha L arabinofurantosidase, beta D gyrosylpyranosidase. These polysaccharidase enzymes are released by the colonic microflora. So a product is designed using some glycoside linkage, right? So here it is drug is dexamethasone, and prednisolone is conjugated with with the help of the beta glucoside and beta galactoside linkage. So this is the product. When dexamethasone T1 beta D glucoside, when it comes in contact with the glucosidase enzyme, then this bond is cleaved, you will get the dexamethasone enzyme. Otherwise, this product is unabsorbed in the upper part of the GIT. Another product is the cyclodextrin product. Cyclodextrin product is, as you know, the cyclodextrin, beta cyclodextrin is used for enhancing the water solubility of water insoluble drug. If the water insoluble drug is encased inside the beta cyclodextrin molecule, but if the drug is conjugated at the OH group present on the surface of the beta cyclodextrin, this prodrug will become the colon specific one. So here this is an example of the diphenyl acetic acid beta cyclodextrin conjugate, which is conjugated with the on the surface of conjugate with the OH group present on the surface of the beta cyclodextrin. So when this molecule, this conjugate come in the colon, the polysaccharidase enzyme will be acting one, and then this linkage will be cleaved, and the one molecule of diphenyl acetic acid will get in the colon, and the beta cyclodextrin, released beta cyclodextrin is excreted with the feces. Another product is the dextran. We use the dextran for the preparation of the product with the dexamethasone and methylprednisolone. The dexamethasone, glutarate dextran ester product, and methylprednisolone succinate dextran ester product. These are the esters, succinate and glutarate on is used for the conjugation of the drug with the dextran, and this conjugate is exclusively release the drug dexamethasone or methylprednisolone by the action of the dextranase enzyme that is released by the colonic bacteria. And site-dependent nanonuclear system, that site-dependent is nothing, it is only just reducing the size of the drug delivery system to the nano level. That will improve the colonic residence time, which will come to the colon. So by filling in the capsule, we can deliver it to the colon. So that will improve the colonic residence times, exerting an EPR effect, and also preferential uptake is possible of these particles by immune cells at the inflamed regions. Avoid rapid carrier elimination by the diarrhea, because in case of the diarrhea or inflamed bowel disease, irritable bowel disease, there are chances of the increasing the motions. So because of that, the motility of the colonic membrane is increased, so the clearance of the particle is faster. So if it is in nano size, that we can reduce the clearance of the nanoparticles. Surface charge dependent nanodelivery system, this is another approach by making the charge in nanoparticles, positive charge or negatively charged. If the particle charge particles, because of the electrostatic interaction between the charged nanoparticles and the inflamed cells, there is a addition of the particles with the membrane. First, strong addition of the particles with the colonic membrane. So if it is positively charged nanocarriers, then it will enhance the adherence to the inflamed mucosa of the colon in case of the inflamed bowel disease. It will reduce the nanocarrier clearance when intestine motility is increased. In the IBD, the motility of the colonic membrane is increased, so that will reduce the clearance of the nanocarriers, will reduce the positive nanocarriers, will reduce the clearance of the nanoparticles. Mucus layer thicker in Crohn's disease, so promising strategy for targeting and attention of the drug delivery system. There are certain examples of the cationic nanoparticles and clondronate loaded nanoparticles comprising cationic polymethylmethacrylate depositaries, chitosan functionalized polylactic glycolate nanoparticles. Chitosan impacting coated liposomes can be used. Negatively charged nanocarriers that will also be beneficial for enhancing the viability of the drug in the colonic, enhancing the colonic viability. it will enhance the adherence because it will enhance the adherence with the higher concentration of positively charged protein in the inflamed. In the inflamed region, there is a positive charge protein like eosinophil, cation protein, or transferrin. So your negatively charged nanoparticle will adhere it and increase the residence time of the degrading system. So the effectiveness of the system will increase. Adherence, if you compare the adherence preference with the different charged particles, the negatively charged having the more adherence as compared to the positive charge and neutral nanoparticles. The example of negative charge in nanocarriers is the anionic nanostructural lipid carriers bearing bidonocide. Another approach is pH-dependent drug delivery system. That is as you know that our intestine is having the variation in the pH in different parts of the GIT. The pH of the terminal lumen and colon is generally very high, and hence the coating with the pH-sensitive polymer or designing nano drug delivery system of the pH-sensitive polymer is more beneficial. We can use this approach if we use the pH-sensitive polymer for coating purpose or designing the nanocarriers. So that will release explosive drug into the colon. The pH, that polymer will dissolve at that particular pSa. If it dissolves at the pH more than 6.5, by the time the polymer is dissolved, delivery system will enter into the colon. So Idrazit is the polymer that can be used. Idrazit L, Idrazit S can be used for designing pH-sensitive drug delivery system for exclusive release of the drug in the colon. So Idrazit, beside pH sensitivity, they are also exhibiting mucoadhesive. There's a dual advantage that will increase the resident times of the nanoparticles in the or drug delivery system in the colon. So example are Idrazit coated nanoparticles or liposomes. But in this case, there are no major disadvantage in inter-individual or intra-individual variability in the pH is more. Another approach is biodegradable drug delivery system. You know that there are a number of polymers, different polymers, which are degraded by the enzymes exclusively released by the colonic bacteria, say azoreductase or polysaccharides. These enzymes exclusively released in the colon, right? So coating or designing of nanodelivery system with the biodegradable polymer like azoaromatic polymer that will be metabolized or degraded by the azoreductase enzyme, alginate, pectin, dextran that are degraded by the polysaccharides enzyme in the colon. So there is saffron and co-workers prepare azoaromatic polymers using the styrene and hema carboxyethyl methacrylic acid with the divinyl azobenzene as a cross-linker. One day, Gour et al., they also synthesize azopolymer using the hema methylacrylate and methyl methacrylate with the BMAAB as a cross-linker. This is an example of the styrene 2-hydroxyethyl methacrylate copolymer, which is prepared by using the divinyl azobenzene as a cross-linker. We can use either divinyl azobenzene as a cross-linker because this cross-linker is having the azo bond in between the two molecules of the vinyl. Or NN bis beta styrene sulfonyl 4,4-diamino azobenzene. This cross-linker we can use for synthesizing the this polymer, right? So this polymer will be exclusively used for delivering drug to the colon because it is degraded only in the colon. This azo bond will be cleave in the colon. So it will become, by the cleaving of this SO bond, the polymer structure will become porous one and drug will be released. So, but during the synthesis of this copolymer, aromatic polymer, one thing we must have to keep in the mind that the degree of polymerization, we should have controlled degree of polymerization in the presence of cross-linker. If it is excessively polymerized, then polymer will become insoluble. It will not be soluble in any solvent, so that will create a problem in the fabrication of the colon-specific drug delivery system. So, well, you can prepare nanoparticles of aromatic polymers, and these nanoparticles, aromatic polymers containing the drug coated with the filled in the enter-coated capsules, gelatin capsule. It will be intake in the stomach, as well as the intake in the small intestine. But when it will reach to the colon, the azoreductase enzyme will be acting on the this hard gelatin capsule. So aromatic polymer coating is dissolved by the action of the azoreductase enzyme in the colon and release the drug. We can prepare aromatic polymer coated systems. As take a drug, prepare a tablet of a drug coated with the aromatic polymer. Then this will be intake in the stomach and the small intestine. When it will come in the colon, the aromatic polymer coating will dissolve and drug will be released. We can use polysaccharides or different type of the gums for the preparation of the colon-specific drug delivery system. The drug is embedded in polysaccharide matrix like gum. In the gum, right, compress it, make a compact mass, and it will be intake in the stomach and small intestine. When it come in the small intestine, the polysaccharide, use polysaccharide is little bit swell, and because of swelling, the sum of the drug may release. And but very little amount of the drug will be released in the upper part of the GIT, or that is in the small intestine. But it will delay the release of these polysaccharides. Gums will delay the release from the compact mass. But when it will reach to the colon, this mass reaches to the colon. The polysaccharide enzyme will degrade the polysaccharides of gum and release the drug in the colon. This is about the studies that about the microspheres that we have prepared here using the pectin as a polymer polysaccharide, right? It's a biodegradable pectin we have used. We have prepared calcium pectinate microspheres. So the pectin solutions dissolve in this PBS, right? And 381% was added. Drug was dispersed, mixed well in the pectin solution. And this solution, pectin solution, is properly dispersed in the isoactive, having the span 80 as an emulsifier, right? And whole emulsion is stirred at 1500 RPM for 10 minutes. We'll get the water and oil emulsion, and the water globules, which are the pectin globules, right? Further stir for this emulsion, further stir for 10 minutes. And this dispersion, will add dropwise calcium chloride solution. So that will calcium ions will react with the pectin and will get the calcium pectinate, which is solid in nature, insoluble in the water, right? So we'll get the calcium pectinate microspheres, wash it with the water, separate it, wash it with water, This is the same photograph of the calcium pectin microspheres without drug. This is with the drug. And then we have performed different studies using the uh, differentiate just to ascertain that the prepared calcium pectin microspheres releases drug in the colon or not. So we have performed in vitro drug release resolution study or drug release in different gastrointestinal fluids like gastric fluid having pH 1.2, simulated intestinal fluid having pH 7.5, mixed simulated gastric intestinal fluid, right? And simulated colonic fluid having pH 7. Yeah, it is containing some fecal content also. So, since pectin is degraded only in the presence by the pectinase enzyme that is released in the colon, so the release of the drug in the upper part of the GIT up to the ileocecal region is very less. Only the drug release is due to the little amount of the drug release is due to the swelling of the calcium pectin microspheres in the colonic, in the small intestine. Right. So when these microspheres will reach into calcium, reach into the colonic fluid or come in contact with the colonic simulated colonic fluid, there is a digestion of the calcium pectinate and release of that drug. So high amount of that drug we observe after the 8 hours. Then we have performed in vivo studies after the administration of the calcium pectin microspheres through the oral route to the agglomerate and dissected at different time interval. We isolated the gastrointestinal tract and separate different parts of the GIT like stomach, small intestine, and the colon, large intestine. And we find that after the 8 hours, most of the drug will absorb in the colon. Similarly, with polysaccharide like guar gum can also be used for the preparation of the colon-specific drug release system. Guar gum, you know that it is hydrophilic in nature, swell in the cold water, forming viscous colloid dispersion. And because of the gelling property of the guar gum, the release of the drug from the dosage form as well is retarded. It is susceptible to degradation in the colonic environment by the polysaccharides enzyme, which are released exclusively in the colon by the colonic microflora. So we prepare cross-linked guar gum microspheres. So guar gum solution disperse dropwise into the castor oil having span-80 and anti-foaming agent at 60 degrees centigrade. Stir it for 10 minutes. So we'll get the dispersion of the guar gum in the castor oil, and the guar gum globules are rigidized by using the cross-linker like glutaraldehyde, toluene saturated glutaraldehyde, and stir it for further 1 hour. We'll get the solid rigid guar gum microspheres, right, which are separated by centrifuge, wash it, and we can use it. These are the same photo micrograph of the guar gum microspheres without drug, and this is guar gum microspheres with drug. Similarly, we perform different studies just to ascertain that guar gum microspheres release a drug in the colon, exclusively in the colon or not. So we perform different studies at different pH. Apart from that, we also perform what will be the effect of the cecal content, colonic content on the release of the drug. We observe that as on increasing the concentration of the cecal content, means colonic content in the dissolution media, the release of the drug from the guar gum microspheres will increase from 31% to 47 and 61%. So we observe mostly 16 to 30% enhancement in the drug release if the cecal content is used in the dissolution media, simulated colonic fluids. Similarly, we use cecal content, which is having the cecal content after 4 days of enzyme induction. Means for the degradation of the guar gum, you will require polysaccharide, which is being released by the colonic bacteria exclusively in the colon. So we induce this enzyme induce the release of these enzyme by the colonic bacteria by feeding the animal continuously for 4 days with the guar gum, plain guar gum. So for the digestion of the guar gum, the colonic bacteria will release polysaccharides. So the concentration of polysaccharides will increase. So if this is being added in, this cecal content is added into the simulated colonic fluid, then the release of the drug from the guar gum microspheres will increase 21%. Further increase 21%. This is another approach where redox drug delivery means redox sensitive drug delivery system, oxygen sensitive drug delivery, ROC sensitive drug delivery system. In case of the inflammatory bowel disease, reactive oxygen species level is abnormally increased. About 10 to 100-fold increase in mucosal ROS is observed in ulcerative colitis. So if you use the polymer thioketal, that is the poly-1,4-phenylene acetone dimethylene thioketal. So we prepare nanoparticle of this polymer, which is ROS sensitive one, and that for the delivery, this is used for the delivery of siRNA, and it will exclusively degrade in the by the ROS. That level is high in the colon, or in case of ulcerative colitis or inflammatory bowel disease. Say active targeting that provides selective drug accumulation at disease site within the colon using the ligands. It will improve therapeutic efficiency, reduce adverse reaction interaction between the targeting ligand and specific receptor expressed on the colon disease sites. So that interaction is strong if the ligands are used, disease-specific ligands are used. Targeting moiety or ligands that can be used, especially in case of the colorectal cancer, that is the transferring folic acid monoclonal antibodies. Number of nano carriers we can use, right, for the preparation of such drug delivery system. So here the liposomes is liganded with the folic acid, right, using the carbamide chemistry. And this is the folic acid coupled liposomes transmission electron microscopy photograph. This is the containing 5-fluorouracil with uncoupled. This is uncoupled liposomes, and this is the coupled liposome. Means surface on the surface of the liposome, folic acid was coupled, and then encapsulation of liposomes in alginate beads. These liposomes are encapsulated in the alginate beads. So liposomes using this method, liposomes to be encapsulated, that is about 10 milligram, containing 10 milligram drug, is first penetrated by the centrifugation, then resuspended in distilled water, then mixed with the stock solution of the sodium alginate, and this liposomes containing sodium alginate solution was dropped at the rate of 10 ml per minute through a syringe nozzle 22 into a 50 ml calcium chloride solution. Then this calcium alginate beads will react with the calcium ions and will get it will convert into the calcium alginate, which is solid in nature. So we'll get stirred slowly for 1 hour to cure calcium alginate gel beads. Wash it with deionized water, dry to a constant weight in the vacuum desiccator. This is the same photograph of the uncoated beads capsulating liposomes bearing oxalating, and this is the photograph beads of the beads containing 5-fluorouracil. And then these beads, which are containing the surface modified liposomes, is further coated with the pH-sensitive polymer like hydrogen S100. So we prepare the solution, milky solution, latex solution of the hydrogen S100 using 5 molar ammonia and stirring for 1 hour. And triethylene citrate was added and stirring was continued for next 1 hour. Anti-coating dispersion was filtered by passing through 0.3 millimeter sieve and sprayed through nozzle on beads. So we prepared this anti-coated, coated calcium alginate beads containing surface modified liposomes bearing anti-cancer drug. This is a schematic diagram. So how it will act. Say liposomes, this is the liposomes bearing the anti-cancer drug. Coupled, the surface of liposomes is coupled with the folic acid. This is the surface modified folic acid coupled liposomes bearing anti-cancer drug. This is entrapped in the calcium alginate beads, right? And these beads are further coated with the hydrogen S100. So this whole system will be intake in the stomach and the small intestine, right? When this, but in the small intestine, the distal part of the small intestine that we at ileocecal region, the coating, hydrogen coating is dissolved, right? And the plain this calcium alginate beads, by the time these calcium alginate beads will enter into the colon, where the polysaccharide enzyme will degrade the calcium, calcium alginate beads, right? And due to the digestion of calcium alginate, the surface modified liposomes will come out, and since these surface of liposomes is ligated with the folic acid, so it will be, these will be taken up by the folic acid receptor overexpressed on the surface of the colorectal cancer. So this targeting, effective targeting of the anti-cancer drug is possible by using this strategy. And this is on this strategy, I received the Visitors Award, that is the President Award by the President of India in 2018. So this is about the cross-linked cytosine microspheres. We use the dispersion method for preparing the cytosine microspheres, right? Rigidize the cytosine microspheres by glutaldehyde tolerant saturated tolerant glutalate saturated tolerant solution, right? And then cytosine microspheres, since these microspheres are quite adhesive in nature, so delivery through the root is difficult one. It will adhere at the upper part of the GIT. So we have anti-coated it with the anti-coated polymer, right? So cytosine microspheres are coated, anti-coated with the anti-coated polymer using the emulsification method. Rinse it after the coating. These microspheres were centrifuged. Rinse it with the N-hexane and dry in vacuum desiccated for 48 hours. This is same photograph of crossing cytosine microsphere without drug, and this is the same photograph of the cytosine microspheres bearing the drug. And again, different studies were performed just to ascertain that how much drug is released in the colon in the presence of the reticular content or in the colon when delivery system will reach into the colon. So because chitosan is degraded by the polysaccharides enzyme release in the colon, right? So the most of the drug is released in the colon. So different study, dilution studies or release studies were performed without the reticular content, with the reticular content, and the reticular content after 5 days enzyme inductions. Enzyme induction, again, it will have the more concentration of the enzymes. So that cecal content was added into the dissolution with a simolytic colonic fluid and perform the dissolution studies. And we find that 26 to 29% enhancement in the drug release if the enzyme is induced after the 5 days feeding of the chitosan. Next approach is the nanocomposite system. As I told you, we prepared the nanocomposites of chitosan, and chitosan has been intercalated in this approach via ionic exchange by negatively charged montmorillonite. Right, chitosan montmorillonite nanocomposites remarkably reduce the ulcer index as compared to the chitosan nanoparticles and the chitosan montmorillonite nanoparticles. And these are highly effective in case for the reduction of the inflammatory bowel disease. So we know that montmorillonite shows anti-inflammatory activity by absorbing the luminal antigen. It will absorb the luminal antigen, increase of the colonic mucin level, and possibly a direct modulating action of the cytokine production by the mucosal cell. It is well documented that epithelial barrier defect induced by the pro-inflammatory cytokine has been restored by the montmorillonite. So this nanocomposite is highly effective, containing the this mesallagine. It's a complete. It's a highly effective nanocomposite for reducing the inflammatory bowel disease or ulcer index. Dendimer, as I told you, in the skeleton of the dendimers, whether you use PAM power or PAM pH dendimers. So in the skeleton, during the synthesis of the dendimers in the skeleton of the dendimer, if we produce that azobond, right, that will be, that dendimer will be colon specific one. It will degrade only in the colon and release the drug. So the dendimers can also be used for colonic delivery of the drug for the treatment of the diseases associated with the colon. Last approach, that is the time release approach. We can use, as I told you, by delaying the release of the drug until it enters into the colon. We can design colons, or we can deliver drug to the colon, right? The first formulation introduced based on this principle was pulsing cap, that was based on the delayed release. It is a similar to hard gelatin capsule. As a hard gelatin capsule, the body of hard gelatin capsule is water insoluble, coated with the water insoluble polymer containing the drug, and the mouth of this body is capped with the hydrogel. Hydrogel may be of the different gums, of different length. The length of these hydrogel, we have to monitor it in such a way that will delay the release. And then this body is capped with the the cap is coated with the anti-coated polymer. How it will act? Say this is the pulsing cap. When it it will be intake in the stomach, but when it will come into the small intestine, the entry coating of the cap is dissolved, right? and the system, the body will expose to the small intestine fluid, right? That is the aqueous fluid. And when the fluid, small intestine fluid, come in contact with this plug is a hydrogel plug or plug of the gums, it will start swelling, right? It will slowly swells, and slowly swelling of the hydrogel leads to lactant that is predetermined by the length of the hydrogel. So at what time it's supposed to expel out, and we have to monitor it, right? And so we design in such a way that it will when this system will reach at the ileocecal region, the plug will be expelled out due to the swelling end. By the time your delivery system, your capsule body will reach into the colon and release the drug. So this is how the pulsing cap is working. Thank you very much. This is the picture of the award, most prestigious award that I received at Rashtrapati Bhavan on 2nd May 2018 on the approach, that is the colon specific drug delivery system designed for the treatment of colorectal cancer using the active drug targeting approach. So we believe, we Indian believes in let us not follow a path set by others. Let us set a path for others to follow. Thank you very much.
Thank you, sir. Thank you so much, Sanjay sir, for delivering such a compelling and insightful session on a nanotechnology-based clone-targeted drug delivery, a new paradigm in a pharma research. Sir, your deep expertise and clarity in explaining the complexity and innovation in the nanocarrier system, targeting mechanism, and their clinical relevance have truly enriched today's discussion. The practical perspective you shared on the research advancement and the future possibility have been incredibly valuable for all the participants. We sincerely appreciate your time, knowledge, and outstanding contribution to the 94th SPSR webinar. Let's go to some QA session. Shall I start, sir? So there are some couple of questions from the audience, sir.
Please.
Sir, the first question from the audience side is, what are the key advantages of using nanotechnology over conventional system for the clone targeted drug delivery system?
Actually, what happened in nanodialysis in my presentation, I have also explained that if you prepare a nanodialysis system, the viability of that nanodelivery system will increase, because if the drug is entrapped in the nanoparticles, the surface area of the for the drug release will increase, right? In case of the colon specific drug delivery system, if we prepare nanoparticles, the residence times, as I told you, residence times of the nanoparticles in the colon will increase. Same time, the EPR effect will also increase. So these are the advantages of using the nanodelivery system. That is why generally I prefer using the nanoparticles for the designing of a colon-specific drug delivery system. Say for active targeting, you will require the nanoparticles or nanodelivery system, because you will require to modify the surface of the nanoparticles, and the modification of the surface of the nanoparticles using the structural techniques. We can use these nanoparticles for targeting the particular part of the colon. I say if you want to target to the colorectal cancer or cancer in the colon, then the surface of the nanoparticle is modified. We can attach some ligands, so attaching is become easy, and these nanoparticles will reach to the receptor site, and it will be taken by receptor mediated endocytosis easily. So these are the advantages of using the nanoparticles in colon specific delivery system. But same time, same time, there are some disadvantages. We cannot take nanoparticles directly through the oral route. We will require some substrate or some capsules by which you have to deliver it to the to the colon. So what I propose that prepare nanoparticles, fill in the capsule, hard gelatin capsule, coat it with the suitable colon-specific polymers, right? So it will be in capsule will be intact in the upper part of GIT. When it will reach to the colon, the coating will dissolve and your surface modified nanoparticles will come out. And since the surface is modified, it is these nanoparticles will be taken up to the receptor site where the suitable receptors are present. So it will be taken up by these receptors in the colon. So these are the advantages of the nanoparticles. Although otherwise we can prepare in simply if you want to deliver metronidazole, you are not need to prepare nanoparticles. You prepare a tablet or you just fill in the capsule, coat it with the suitable polymer and deliver it, because the whole metronidazole you are required to deliver to the colon, right? So there is no need to be taken up by some receptors in the colon, because it has to kill the metronidazole has to kill the amoeba present in the large intestine. So this is
Right, sir.
advantage of the nanotechnology.
So well justified. There is one more question from the participants. So what makes the colon an ideal site for targeted drug delivery in terms of disease treatment and drug absorption?
I just could not get you. Please repeat
Okay.
it.
I'm going to repeat, sir. So what makes the colon an ideal site for targeted drug delivery in terms of disease treatment and drug absorption, sir?
Say, especially colon-specific regulatory system, in my view, it is most suitable for the treatment of the diseases associated with the colon, for local delivery of the drug to the colon, right? Say for the treatment of inflammatory bowel disease, for the treatment of ulcerative colitis, for the treatment of Crohn's disease, for the treatment of the colorectal cancers. These are the diseases which required specific high concentration of the drug in the colon. And right, for the treatment of if it is, as I told you, if these for the treatment of the drug, if the drug is delivered through the conventional dosage form, then it will be reaching to the colon through the blood circulation. So the toxicity of the drug will increase, because before reaching to the colon, drug will be circulating in the whole body. So you will get some of the side effect of toxicity of the drug, right? So if that is why it is become essential to deliver drug directly to the colon. Directly to colon means it should not go to the systematic circulation. It will be whole drug will be should deliver to the colonic lumen for the treatment of these or diseases associated with the colon. Sometimes, say in case of the peptides or say insulin delivery, where absorption of the drug is required, right? Peptides or insulin are degraded in the gut, right? And because of the very complex structure of the this insulin, the absorption from the gastrointestinal membrane, especially from the stomach or the small intestine, is a difficult one. And same time, the drug is, if it is insulin is released in the small intestine or stomach, it degrades, right? If you deliver it to the colon, right, that will save its degradation, and but the transmission of the insulin or peptides from the colonic membrane is not an easy task. You will require to change the permeability of the colonic membrane. So the permeation enhancers you will require to use if you want to deliver peptides or peptidal drug like insulin through the colon route. So if the systematic circulation absorption is required, through the colonic route, you will require to use some penetration enhancers. So in both way, for local disease, colon specific drugs they are giving very good results, right?
So there is one more question, sir. Which nano carrier system might be a nanoparticles, liposome, missiles, which have shown a most promise in the colon targeted drug delivery system, sir?
Say, if you talk about my experience, I'm very comfortable with the liposomes because the liposomes is very potential carrier. But use of liposomes for oral delivery is having some queries, right? Because liposomes, for the fabrication of liposomes, some phospholipids are used. They are more sensitive in the gastrointestinal fluid or lumen, right? That liposomes may degrade, right? But its degradation, we can protect it by encapsulating in the hard gelatin capsule. So in another way, we have prepared nanoparticles of the some biodegradable polymers. Preparation of the nanoparticles is easy as and stability of the nanoparticles is more as compared to the liposomes in the gastrointestinal lumen. So in my way, this nanoparticles are most suitable for the fabrication of the colon-specific drug delivery system because these are the more stable as compared to the liposomes one. And we can use some biodegradable polymer like guar gum, chitosan, chondroitin sulfate, right? Such type of pectin we can use for the fabrication of the nanoparticles and making it colon-specific one.
Okay, so in continuation of this question, I have one more. How do we ensure the stability of nanocarriers during their passage through the upper GIT tract, sir?
Yes, actually, what happens is if we use polysaccharides, gums, right? Or as I told you, liposomes degrade in the gastrointestinal lumen. You are required to protect it, right? By encapsulating in a certain type of the capsules or embedding, immobilize in the some polymer matrix, right? So you are required to protect it from its degradation in the gastrointestinal tract, or by coating with some biodegradable colon-specific polymer, or by the pH-sensitive polymers. Those polymers, pH-sensitive polymer, which dissolve at the pH greater than 6.5. Such type of polymers we can use to protect this degradation in the gastrointestinal lumen. But if you do not coat it, say if you use chitosan microspheres or chitosan nanoparticles, they are very mucoadhesive in nature, right? When you deliver nanoparticles of microsphere powder through the oral route, it will stick to the buccal cavity, right? It's a highly bioadhesive nature. So for just to avoid this bioadhesion in the upper part of the GIT, you will require to coat it with the pH-sensitive polymers or some polymers which will reduce the addition of the chitosan particles, right? Or some gums like gums which are swell in the when these gums or nanoparticles of gums come in contact with the gastrointestinal fluid, which is equational nature, the gums will swell, right? And due to the swelling of some of the drug may release. To protect the release of the gums and their adhesion with the mucosal membrane of the small intestine or stomach, you will require to coat it. So we can enhance its stability by coating it, right? And we can reduce its drug release, interrupt drug release by coating it with some pH-sensitive polymer. So when we design any colon-specific drug, we have to keep so many things in our mind. It should not adhere to the upper part of the GIT. When I say upper part, as I told you, it is the stomach and the small intestine. It should not stick with the intestinal membrane, right? it should not release drug in the upper part of the GIT. So we must have to keep these things in the mind. And same time, you will require, because it's a long time drug has to face with from the different environment in the gastrointestinal tract, from the buccal cavity, then stomach, then small intestine. So different type of the fluids that come in contact with the different types of the fluid. So we have to keep the nature of the fluid which will come in contact with your nanodrug delivery system, right? And we must have to think that whether it is degrading your delivery system, if it is degrading, whether it is having any effect on the delivery system in the upper part of the GND, that we must have to see. And same time, we have to accordingly rectify that problem. So these are the problems we face. And I think this will be for solution for the nanoparticles, develop nanoparticles.
Yes, sir. So let's come to the last question. The question, here we go, sir. How effective are the nanocarriers in improving the treatment outcomes of the inflammatory bowel disease and colorectal cancers?
Your voice is not clear. Can you repeat again?
Audible, sir, now?
No, it is okay, fine. Now.
Okay. So the question was how effective are the nanocarriers in improving the treatment outcomes of the inflammatory bowel disease and colorectal cancer, sir?
Actually, inflammatory bowel disease or colorectal cancer and nanoparticles, as I already discussed, that nanoparticles are having a beneficial effect. It will not only increase the residence time, same time it also increase the EPR effect, right? So we can modify the surface of the nanoparticles by attaching the suitable ligands, which are the cancer-specific one, right? So the receptors which are present over the surface of the cancer, that ligands, specific ligands, we can use when attach on the surface of the nanoparticles, and we can get the target specific delivery, means cancer specific delivery of the your drug. Same time, in case of the inflammatory bowel disease, in the inflammation, the motility is changed, the adherence, the thickness of the mucus is also changed. So there are so many problems. The ROS level is increased. So these can be used as a triggering means for triggering the drug release, for increasing the residence time of the nanoparticles in the colon, especially in case of the inflammatory bowel disease.
Right, sir. So before I going to conclude the session, I sincerely thank you with the folding hands to the respected Professor Sanjay Kumar Jain, sir, for answering all the questions with such a clarity, depth, and enthusiasm. So your thoughtful responses have greatly enhanced our understanding of nanotechnology-based colon-targeted drug delivery systems and their potential in the modern pharmaceutical research. We truly appreciate the way you address each query with practical insights and academic rigor. It has been a privilege to learn from you, your vast experience, sir. Thank you once again for your outstanding contribution to the 94th SPSR webinar, sir. So, my dear audience, as we conclude to the 94th SPSR webinar, we extended our heartfelt appreciation to Professor Sanjay Kumar Jain, sir, highly respected for his exceptional and enlightening session on the nanotechnology-based colon-targeted drug delivery, a new paradigm in the pharma research. Today's session provided a deep insight into the emerging role of the nanotechnology in a site-specific drug delivery, particularly for the colon, for advanced formulation strategies to the therapeutic application and the translational challenges. Professor Jain's presentation has enriched our understanding of how this innovative approach can revolutionize treatment outcomes for conditions such as inflammatory bowel disease and the colorectal cancers. The clarity of explanation and real-world applicability of the concept presented today will undoubtedly inspire continued research and innovation in this domain. On the behalf of Society of Pharmaceutical Sciences and Research, also known as SPSR, we sincerely thank you, Professor Jain sir, for his invaluable contribution and all the participants for their active engagement and interest. We are again deeply thankful to Dr. Shashi Alok sir, the National President of SPSR, for honoring us with his presence. We recognize and appreciate your commitment and guidance, and we offer our profound thanks for such significant input. We would also like to express our appreciation to our convener, Mrs. Monika Sabharwal ma'am, for her pivotal role in organizing this event. We extend our gratitude to all the SPSR team members for their collaborative efforts, which have broadened our understanding and fostered cooperative learning. Your collective contributions have made this webinar a success, and we look forward to continued collaboration in advancing pharmaceutical sciences and research. We look forward to your participation in the future SPSR webinars as we continue to explore groundbreaking topics, foster knowledge, sharing in the pharmaceutical sciences. Thank you once again. We look forward to welcoming you to our upcoming webinars. Stay curious, stay inspired, and stay subscribed. Have a wonderful day ahead. Jai Hind.
Thank you. Thank you, Rishabh. Thank you, Shashi. Thank you, Monika. Thank you all, SPSR team.
Thank you, sir.
Thank you. Thank you very much. Can I leave now?
Yes, sir.